Bueno, vamos a esperar, como buenos españoles, normalmente la gente llega tarde, pero nosotros vamos a empezar. Para mí es un placer presentaros a Markus como nuestro ponente que nos va a ilustrar en el tema de las aplicaciones de los péptidos dentro de distintas patologías. En particular, lo va a hablar de los péptidos para el desarrollo de medicamentos. Tiene atractivo este tema de los péptidos porque, como estábamos comentando anteriormente, las compañías farmacéuticas realmente odian los péptidos como fármacos y no les gusta desarrollar estas moléculas como fármacos, y muchos de los péptidos que han entrado en fases clínicas han sido abortados, o ha sido abortado su desarrollo en la fase clínica 2, no por falta de actividad o de eficacia, sino porque simplemente las compañías farmacéuticas que pueden financiar los estudios en fase 2 y fase 3, no han mostrado interés en este tipo de moléculas, a pesar de que para muchas aplicaciones terapéuticas los péptidos son muy superiores posiblemente a las moléculas orgánicas pequeñas, pensando por ejemplo en aplicaciones para la ruptura o la interferencia de interacciones proteína-proteína dentro de complejos que pueden estar implicados en distintas patologías humanas. Obviamente esa es una área que todavía tiene un potencial de desarrollo tremendo, aun cuando las compañías farmacéuticas no estén mostrando mucho interés, aunque yo creo que ha habido un pequeño cambio en estos tiempos, y las compañías farmacéuticas pues ya también cierto es que gracias al desarrollo de la química de péptidos y a las modificaciones que les han sufrido y que les ha estabilizado y les ha hecho un poco más droga que eran antes, pues están mostrando ya las compañías farmacéuticas pues un interés realmente en incremento con respecto a ellos. Si bien en las compañías farmacéuticas el interés ha sido bajo por los péptidos, en la industria cosmética ha sido todo lo contrario. El interés por los péptidos ha ido creciendo de forma exponencial. Y ha ido creciendo de forma exponencial por varias razones. Una, porque desde el punto de vista de aplicación tópica son unos buenos compuestos cosmecéuticos, y dos, porque la síntesis en la producción de péptidos ha bajado el coste de forma tremenda, lo que lo ha hecho atractivo para esa industria cosmética. Y muchos de los activos cosméticos que hoy en día tenemos, obviamente, provienen de péptidos que están ahí. Después, otro de los aspectos importantes dentro de la formación de Markus es que realmente lo que ha aprendido y lo que más ha aprendido de los péptidos viene del estudio de la diversidad que existe en los venenos naturales. Venenos de serpientes, venenos de escorpiones, venenos de arañas, y en esos venenos hay unos cócteles de péptidos que con una diversidad funcional tremenda, que es lo que realmente ha ilustrado a los químicos a poder imitar toda esa diversidad para poder generar, a partir de todas esas bibliotecas naturales, actividades que estén dirigidas a específicas dianas terapéuticas. Y dentro de esos venenos, uno de los temas de mayor interés, obviamente, ha sido de los canales iónicos, en el cual hemos estado ahí realmente discutiendo acerca de ello. Y me comentaba un caso de una araña, ¿verdad?, que realmente es una araña, este es un caso muy particular, en que hablando fundamentalmente de la inexistencia de péptidos inhibidores de los canales TRP que están implicados en dolor, y eso es lo que trabajamos en el laboratorio, y ya que la mayoría de estos péptidos que tienen las arañas o las serpientes son péptidos que son activadores de los TRP, y el dolor que produce una picadura de estos animales. Pues me comentaba que realmente eso no es del todo cierto, que se acaba de publicar en Science Communications, en donde un investigador, colaborador y amigo suyo, lo que ha detectado es que hay una araña que puede tener dos cócteles distintos dependiendo de cuál es la finalidad. Tiene un cóctel que lo que hace fundamentalmente es, si va a la araña, va a cazar a una presa, realmente lo que tiene es un cóctel de péptidos que hace es fundamentalmente paralizar a la presa y por tanto está formado por inhibidores del sistema nervioso y con lo cual lleva a esa paralización. Y si va a defenderse frente a un ataque de algún peligro, lo que tiene es un cóctel, cambia a un cóctel en donde produce fundamentalmente dolor. Es decir, tienen esa plasticidad para cambiar el cóctel de péptido, bien para producir en una parte dolor o bien para producir lo que es o sea, inhibidores y activadores de los mismos canales son capaces de incubarlo de forma natural. Lo cual resulta interesante, como digo, es un trabajo que han publicado en Nature Communications y que nos comentaba hace un momento. Y yo ya me voy a quedar, si no voy a hacer la conferencia aquí, siguiendo las anécdotas, voy a dejar a Markus, que le agradezco muchísimo que haya venido y que nos ilustre en los últimos avances en torno a la química de ciertos y sus aplicaciones en el desarrollo de veneno. Muchas gracias.
Thanks a lot, Antonio, for inviting me here. And the first time now it happens, I'm quite keen to explore the city afterwards. And it was a spot on introduction in Spanish. I wish I could do the same in Spanish. I understood most of it. I'll get there in a couple more years. I hope I can fill in with a couple of more technical details and in general details of what we do with peptide drug discovery or peptide drug development in general. Particularly, I would like to focus on one approach using selenocysteine as an amino acid to improve methodology and stability of these maybe future peptide drugs. So let me start with giving you a little bit of an overview of the peptide drug field on a global scale. Peptides get more and more attention lately, not only as pharmacological tools, which we all know these are great due to its potency and selectivity, but also as peptide drugs. However, when we look into the global drug sales, we can see the peptide drugs are a very small minority, only making about 3% of the total current drug sales on the market. And from this 3%, insulin takes like 60% of all the revenue. So we still have a long way to go in terms of marketing peptide drugs compared to small molecules or even biologics. Biologics themselves is a very recent field, like 10-15 years ago, and they overtook the peptide field, who is much older by far. But as Antonio mentioned, the mindset of the pharmaceutical industry is changing, and more people investigate peptides as therapeutics. Examples are like Amgen, Ironwoods, also Genentech moves in, and more companies move into this space. When we look at analyzing these peptide drugs on the market, most of them are agonists. Now, this is not a rational decision per se when it comes to drug development, but derives that the first generation of peptide therapeutics derives from human hormone analogs. I'm talking about insulin, oxytocin, vasopressin, serotonin ones. These are all peptide agonists, peptide hormones, which play functional roles in us humans. And when you have a deficiency, the first approach was to just replace them by in the early days it was extracts from beef, like cows and porcine. Later they used recombinant methodologies to produce them. And insulin today is still produced recombinantly. A couple of decades later, they realized that nature itself has a much higher diversity of these peptide analogs, and that some of these peptides in nature fare much better metabolically and pharmaceutically than actually the human analogs. Two prime examples would be Byetta, which is like a venom peptide extracted from the saliva of the Gila monster, and that's used as a GLP-1 agonist. We have, GLP-1 agonist in humans has a half-life of 2 to 3 minutes, and many pharmaceutical industry try to, get the human analog into the clinics, into the market, and they fail fairly extensively. However, that venom peptide from the Gila monster turned out to have a much higher half-life, for hours, and is now an injectable used for diabetes. Generation 3, we mentioned rational design. Now, rational design, we still lack a lot of it. Like most of these compounds on the market are either derived from nature or human analogs. So there's only Fusion and the concept of decoy as a therapeutic approach. And then there's a lot of medicinal chemistry done that, for example, pasado, which has been approved in 2012 from Novartis, where classical medicinal chemistry take somatostatin and then make it more rigid, cyclize, improve the half-life. So a lot of classical medicinal chemistry is being done on peptides. However, we argue that this might work in many cases. However, the generation 4, the future generation of peptide therapeutics, will not be in rational design or medicinal chemistry alone, but will come from efficiently sourcing natural sources. We are particularly interested in venom peptides, but another approach would be different display technologies such as phage and RNA display technology, where you can evolutionary in petri dishes or in vitro generate and select for binders, peptidic binders. You can see a trend going from very simple and flexible peptides to more rigid and more stable and cyclized peptides. And you find the same trend in venom peptides. Many of these venom peptides have about hundreds of millions of years of evolution being fine-tuned and selected for. And you can see that many of these have rigid scaffolds, which are stabilized by disulfide bonds, that then maintain the secondary structure where you find the pharmacos. And this is responsible for the really high potency and selectivity. So in the next couple of minutes, I really want to move into details of venom peptides, how they are composed, where do we find them, and how do we discover and develop them into pharmaceutical drugs. I'll start off with an unbiased ligand discovery, where we actually go out in nature and look at diversity of venom peptides and then screen them against targets. And then in the second part of my talk, I'll focus on one receptor target and then see how we can use evolution or nature and find more potent or more selective ligands for these targets. So let me start that venom peptides work, and some of them are already on the market. It all started with the ACE inhibitors. These are on the dipeptides, so we don't really include them as proper peptide chemistry, but they are good for hypertension and congestive heart failure, and they initially extracted from the Lancet vitamin. I already mentioned Byetta. It's a GLP-1 agonist, about 39 amino acids, no disulfide bond, comes from the Gila monster. And Combus markus, is like a marine predatory cone snail, has an anti-voltage-gated calcium channel blocker, venopeptide. And this is in the clinic for treatment of severe chronic pain. And here you can see already it becomes more rigid, more structured, free disulfide. And this is very much, I think, the trend where we're going to be going. There's really no better place to study venopeptides than Australia. When you go there, and I think many of you have been there, anything that you touch can potentially kill you. And I was very lucky to have been working with Professor Paul Elwood at the Institute for Molecular Bioscience in Brisbane, which is a leader in venopeptide drug discovery. And when you join his lab, every student gets to choose an animal that is venomous, and you study the venom. These ones are all a couple of animals we started. We stopped looking at snakes since they became they're very fast and they're vicious, and after we had like an incident, we stopped researching venoms of snakes, and we moved to a few slower animals. I thought I was very smart of choosing the cone snail when I started there. My priority was safety, coming from Austria, so I choose it because it's not the fastest predator, and I think I can handle it. The second criterion of why I chose it is like they usually hang out in the places where I like to hang out, which is mainly subtropical islands on the reefs, and this is where they hunt other mollusks or fish. They have enough true venom to work with. We have other PhD students who, for example, study the venom of ticks, and they have to collect thousands to 10,000 to get enough venom to work with. So I definitely did not want to do that. And they have a very good biodiversity. There's about 500 to 700 of these species of cone snails, and each species has usually a unique cocktail of venom, and in one of these cool venoms you find like hundreds to thousands of individual bioactive peptides. So this is a very conservative estimate of how many toxins are out there, and we believe this goes probably much more, over 100,000. And this is hardly untapped. So I think we have about 500 peptides characterized so far. So this is pretty much nothing. Being a chemist, I also like the chemical accessibility, like how, what can I synthesize rapidly. This is really important when you do structure activity relationships. It's a difference if you have a 40 or 50 male to make 100 analogs, or you only have a 12 male to make 100 analogs of. So when you look at the venom of Marmoris, you see that the main diversity is around 2 kilodalton. When you look at spider venoms, this peaks rather by 4 kilodalton. So I think chemical diversity-wise and accessibility, the consonant has the better access. How do these chronotoxins or venom-peptides look like in general? As I mentioned before, they all have these rigid disulfide bond frameworks, and these frameworks stabilize secondary structural motifs such as beta turns, alpha helix, beta sheets, and they also call often mini proteins because they're usually very small in size but have the same secondary and sometimes even tertiary structures than proteins. The chronotoxins are usually produced in the venom bulb and then delivered into the venom duct, where it's then loaded into this harpoon-like needle, which is then injected in either prey or against defense from predators. They are very potent and receptor selective, and they are a main tool for electrophysiology to differentiate different ion channel subtypes. People often ask me how selective are they? We did this little GPC paper on just 2 isoforms of the same peptide, which is alpha chronotoxin A or B. And it turns out when we have the 1,3,2,4 disulfide connectivity, we have this alpha helix. And this antagonizes the alpha-3 beta-4 nicotinic acetylcholine channels. However, in these alpha-3 beta-4, you can have different subunits stoichiometric. And when you switch to disulfide conformation, you have like a competitive antagonist that only antagonizes the alpha-3 beta-4, where you have 2 beta units and 3 alpha units. So you can use these not only to differentiate different subtypes of an ion channel, but you can even differentiate within the stoichiometric composition. They're also really interested from a chemical scaffold point of view. So you have these alpha-chronotoxin BC1.1, which made it into phase 1 clinical trial as an analgesic, and at that stage they thought the target was the alpha-9, alpha-10 nicotinic acetylcholine receptors. Turned out then that in humans the alpha-9, alpha-10 is only expressed in the earlobe, and people ask the question like, how can an antagonist of the earlobe be anywhere as a therapeutic analgesic? So people started questioning the actual target of the nicotinic acetylcholine receptor, and then later they found out that the same scaffold and the same peptide, which antagonizes the nicotinic acetylcholine receptors, agonizes also nanomolar potency the GABA B receptors. And this is just phenomenal when you think about it, that this 15, 16 amino acid scaffolds can block a nicotinic acetylcholine receptor and activate a complete different structurally unrelated receptor, which is the GABA B GPCR. But of course, from the constant perspective, it doesn't matter, because if this paralyzes the prey and this paralyzes the prey, of course, it makes sense to only create one scaffold that does both things. And when you look at like 600 million years of evolution, you understand why these scaffolds have been like selected. So we looked into other places and different scaffolds to see if this is like a general theme. And we found Bangawa toxins, for example, have this 3-finger motif, and again they are able to mutate these loops to target different receptors, and this includes the nicotinic acetylcholine receptor, the muscarinic, and then again muscarinic and then GPCR. We also find mastoperan, which is like a cell-penetrating peptide. At the same time, it interacts with G proteins inside the cells. And then we have protein 1A, which also blocks the adrenogenic receptors. So for people who've never seen a corn snail in action, like me when I moved to Australia, this might be a very fun and interesting one. So this is Cornus, who is hunting fish, and you can see once it's injected the harpoon, it paralyzes the fish pretty much in milliseconds, and then it just pulls it in and digests it as a whole. And this is pretty much how they hunt at night. Now, how do we go about to actually discover these venom peptides? The traditional approach was to go out in the reef, go in the jungle, find the spiders, find the snails, and then we pretty much milk them or dissect them, and we get the crude venom out. Once we have this one, we purify them on reverse phase HPLC columns, fractionate them, and then screen the fractions, which each fraction still has like hundreds of peptides in there, against selected therapeutic targets. This goes through like flipper screens or electrophysiology assays, depending whatever target you're after. Now, in the last 5 to 10 years, there's been like a huge paradigm shift on how venom peptide drug discovery is happening. With next generation sequencing and genome and access to transcriptomes, what you can do now is you just analyze the transcriptome and the genome and get like thousands of sequences out, and then you cross-reference it with advanced proteomics to see what is actually expressed in the crude venom. And then you use bioinformatic tools to cross and correlate these. And then once you have the sequences, you can then decide if you want to express them, clone them in vectors and express them, or you synthesize them using chemical synthesis tools. Once you have the peptides, you then do NMR structure analysis and start with your SAR studies in terms of like optimizing them or looking for the right targets. Now, it has advantages and disadvantages. I think the main advantage is it's very rapid, and the advantage of genomics is it's unbiased. So when you look at compounds which are like overexpressed in the venom, this is like the ones you pull out. But that not necessarily means these are like the most potent ones or most selective ones. Because the more potent and more selective one, you don't know, the corn snail doesn't have to express in huge quantities. So using the genomics approach, you get like an unbiased approach of getting all this sequence information. However, what you don't get is like you don't know the target. Yeah? So the disadvantage are also you lose post-translational modifications. People are not going out and live on the reefs anymore, so everything can be done in the laboratory. And you also miss out on the target-guided selection. Having thousands of sequences puts a lot of pressure on the synthetic and the high-throughput screening in terms of. Now, with peptide synthesis itself, the assembly and synthesis of these peptides is highly automated, so that's not a problem. But the real problem is like the diisotope. When you synthesize a peptide, you get it in reduced and linear form. And then when you oxidize it, it can form different diisotope isomers. These different diisotope isomers have all the same mass, the same retention time on HPLC, but only the active, the endogenous, has the bioactivity. So this becomes a very tedious, time-consuming effort to either do NMR or reductive alkylations or activity studies to find out which ones of these is actually the endogenous one. So the way people solved this was using orthogonal cysteine protecting groups. So they can actually direct the folding into the one that they know is the endogenous fold. However, using orthogonal cysteine protecting groups means like extra purification steps, extra deprotection and folding steps. So it's not a really efficient approach if you want to create like a library of 200 of these, for example. When it comes down to 3, 4, or 5 dozen ones, this becomes even more complex. You get like 50 and 105 different isomers, and it becomes like a nightmare to really find out which one is your active one. So we decided to tackle this problem using quite a novel approach, using selenocysteine instead of cysteine. And this has been work leading up to, particularly by the group of Luis Maruda, who really characterized selenocysteine versus cysteines and looked at the similarities and differences of these two amino acids. And when you look at the physical chemical properties, you see that selenium and sulfur are very similar. And people have used selenium as an isosteric replacement to sulfur in many studies. For example, one of them in X-ray crystallography studies, because selenium is a heavy atom, so they implemented selenium instead of sulfur to solve the phasing problem. Yeah? Selenium itself has like other properties as well, like there's 6 stable isotopes, so when you do mass specs, you get like this hump, so you can track selenium in peptides using mass spectrometry. Selenium-77 has a spin of half, so it's also NMR active, so you can do selenium NMR with it. And in general, selenium in form of selenocysteine is a better nucleophile and also a better leading group than cysteine. However, what I want to focus here now is when you look at the redox potential and the pKa, you see that selenocysteine has a lower redox potential and also lower pKa. And we wanted to take advantage of this lower pKa and lower redox potential because it suggested or indicates that when you have 2 selenocysteines and 2 cysteines in one peptide, you have a preferred disulfide formation over disulfide formation. And we wanted to exploit this concept to fold these peptides into the right conformation depending on where we place the selenocysteines. To do so, we need the building blocks, and this slide should just show you that all the protecting groups of cysteines are also available for selenocysteines. Some of them are published, some of them are commercially available, but all of them can be synthesized in a lab with very little organic chemistry knowledge within a week and really big scale. So we used bulk chemistry. So our main animal was this oxalinocysteine protected with methyl benzo. And we can make 30 grams in 4 days, and that usually lasts for a year. And we hope that this is commercially available soon. So coming back to the synthetic scheme, so as I mentioned, we now place pairs of selenocysteines into these condotoxins, and then once we cleave them, the disulfide bond forms first, and then you increase the pH, and then you form the disulfide. And if you place them in a different conformation, like here the 2,4, you can drive this one to the same different isomers. And therefore, you can elegantly take care of not using orthogonal cysteine protecting groups, but just using selenocysteines in the one-step approach. When we look at how this looks on an H+C, here you see that all selenocysteine all cysteine are A1B, and when we fold it, we get both the ribbon and the global amount, 50-50. When we put the selenocysteines in the position we want, we drive this equilibrium to the desired one, and we always know which fold we have. When we do the other side, we can push it towards the ribbon at about 80 to 90%. Now we extended this methodology to many different alpha contoxins and also to free disulfide ones. And observations we make is like it works really fast and efficient. For example, let's start here. If we have A or B, all the cysteines at pH 5 over 24 hours, nothing happens, it stays reduced. However, if we have the selenocysteines present at pH 5, within 2 hours, it's fully folded. The reason is the disulfide bond form at low pH already, and then bring the 2 cysteines into close proximity, and then lowers the activation energy to form that disulfide bond. If you want to just do it standard in pH 8, within 10 minutes, it's fully folded. You can see here, the percentages, like it really drives it to wherever fold you want. One other observation we did is when we cleave these peptides off the resin using hydrogen fluoride or TFA, the disulfide bond forms instantaneously. Literature, however, suggested that the pK is around 5.2. So we were never able to isolate the reduced selenium, the selenium. So we were questioning the literature values of this pK. So we elegantly took selenium-77 NMR and titrated the hydrogen selenium over pH from 0 to 10, and pretty much were able to determine the pK, the exact pK of these selenocysteines within peptides for the first time, showing that first the N-terminus is a much lower pK than the ones within the peptide, and both pKs are much lower than the literature value. So when you have pKs around 3, it makes much more sense that the disulfide bond forms at lower pHs than if you have a pK of 5. Another question that's solved there is when we have about 20 selenium proteins in our bodies. And in most of these selenium proteins, selenocysteine is not incorporated in the form of a disulfide bond, but as a single selenocysteine. So if we have such a low pKa, we question why wouldn't you see these protein oligomers and forming disulfide. So we looked at crystal structure of two of these selenium proteins, and we found that the selenocysteine, the negatively charged one, is actually stabilized in catalytic triads of these enzymatic pockets. And it's pretty much stabilized by the lone pairs of these nitrogens. Same with selenocysteine. Our final goal was not only to fold them in solution, but to make libraries on resin, make 100, 200 of these alpha compounds. So we moved this system onto resin, and to do this, we needed a special resin, which pretty much swells in organic solvents to synthesize and assemble these peptides, but then to deprotect them in HF and then move them in aqueous solvent to oxidize and fold them while still attached to the resin. The way we did it is chematrix is like a polyethylene glycol-based resin that is amphiphilic and swells both in water as well as in DMF. And we use the safety catch acid label linker, scour linker. In its oxidized form, it is stable to TFA, and you can assemble all the peptide. It's also stable to HF, so you can completely deprotect it while the peptide is still on there. And then when you reduce it with ammonium iodide, you get rid of these oxygens, and then it becomes acid labeled, and it comes off the resin. As you can see here, just VC1.1, all system ones, falls nicely into the major active isomer. When we do the all system on resin, the resin itself, the environment has an impact on folding, and you get all three isomers. However, again, once we put the selenocysteine in there, we can reverse that isomer formation, and we only get the one desired isomer. So our selenocysteine methodology also works on resin. And with that, we were able to synthesize 50 to 100 of these alpha compounds. We generally just purify them, not individually on one H+C column, but we can go up to 10 different peptides on a single run and purify them in enough quantities for assays. We were also interested if, once you have this disulfide bond in the peptide, do they behave identical to the old cysteine ones? So we did electrophysiology studies on the nicotinic acetylcholine receptor, as well as rat diaphragm studies. And we showed that most of these selenium analogs behave identical than the cysteine analogs. However, one or two, we found actually tenfold more potent than the native cysteine ones. So we wanted to understand why this is. And we looked into NMR to see if the secondary structure of these selenium analogs are identical to the cysteine ones. As you can see here with CD and alpha shift NMR, they are nearly identical. We were also really lucky to get a crystal structure of the old cysteine ones with the disulfide bond, and when we overlay them, they are nearly identical. So the secondary structure, in our opinion, has more impact on these potencies. The only difference you can see here is the disulfide one is 0.3 actually longer than the disulfide one. But the torsion angles pretty much compromise these again, and there's really no differences in secondary structure. So we finally found out what caused the more potent activity, and when we look at this reverse phase of the C18 H+C column, the potent selenium analog eluted later, suggesting that it's slightly more hydrophobic than the old cysteine ones. So we went back to modeling. This is the nicotinic acetylcholine binding protein, and when you look at the binding pocket itself, in orange it's a very hydrophobic pocket. So we think that the slightly larger disulfide bond and the slightly more hydrophobic carrier of the disulfide bond is the main reason why this binds more potent onto this pocket. We also looked into stability, particularly since a lower redox potential of the disulfide bond indicates that it will be harder to reduce than a disulfide bond. And we're particularly interested in this scrambling approach because when you look into targets which are intracellular, where you have micromolar concentrations of glutathione in them, If this is, for example, your drug, and it goes intracellular, the glutathione will, within minutes, break this bond and shuffle it into this inactive one. So not only like enzymatic degradation is a problem, but also interaction with free thiols can deactivate your peptide drug. So once we place the disulfide bond in there, we can suppress this scrambling and improve the half-life by doing this. So in my second part of my talk, I want to talk about specific ligand discovery. So we're still in the realm of natural peptide, but this time we focus on a single receptor and we want to find selective targets for these receptors. Yeah. The receptor we chose is the oxytocin or vasopressin receptor. The reason I chose it because we found vasopressin-like peptides in the venom of the coulson. And for me that was puzzling. Like, why would you want to have the LAF molecule in as a venom peptide in a fish hunting coulson? So that's still very puzzling. The other reason I was interested in, like, these disulfide-rich peptides have been well characterized, particularly towards ion channels. Now, these non-disulfide-rich, which still is about 40% of all the venoms of the common toxins, are hardly characterized. And some of them target transporters and GPCRs. And we all know that GPCR is a great drug target. So I thought maybe we could combine the diversity of venom peptides with targeting GPCRs. So I started reading up on the oxytocin and vasopressin field, and when you start reading it, you realize a couple of things. First, it's a very old field. Oxytocin is like the first peptide ever synthesized and discovered. And there's 4 GPCRs, and they all have enormous extracellular homology. And that is a big problem for translation of pharmaceutical development of these receptors. Because even oxytocin acts on all of these receptors, and vasopressin also acts on the oxytocin receptor. So even our endogenous ligands are not selective. So selectivity in us humans is modulated not by the ligands itself, but more by upregulations or enzymes, etc. And oxytocin and vasopressin are very old signaling systems, so they are present everywhere. They're present in the gut, in the blood, in the heart, in the CNS, and they have lots of physiological, very important function. This goes from like reproduction to water household to blood regulation in terms of pressure. So there's a huge need to find selective agonists and antagonists of these 4 subtypes. And pharmaceutical industry put a lot of money in there to find smaller molecules to target these ones. And many of them failed in phase 2 and phase 3 clinical trials. So far, we don't really have a single small molecule drug for these receptors. And there's plenty of applications therapeutically. So while there's been a lot of standard classical medicinal chemistry being done, particularly from Maurice Manning and Victor Ruby's lab, and they've identified a couple of really good antagonists as well as agonists, it was not so successful as we expected, and we still, 60 years after the discovery, have not a complete toolbox of these selective agonists and antagonists. So we decided to look at this from a very different point of view. And when we look at the evolution of these oxytocin vasopressin receptors, we see everything leads back to this ancient vasotocin. And then 600 million years later, oxytocin vasopressin in humans, it looks still the same. We still have these nonapeptides, system between position 1 and 6, type 2 beta term, type 3 beta term, which is like very important for the binding of these receptors. And we find these signaling systems everywhere. We find this in water flea, in octopus, in kangaroos, in humans, and in the venom of the common constants. So we decided, or we argue that if we find these ligands and synthesize them, we have a much higher success rate in terms of finding selective potent oxytocin analogs than doing random combinatorial chemistry or medicinal chemistry screening, particularly because nature preserved these type 2 beta terms and type 3 beta terms. So we created this evolutionary tree to track down which, or in the literature, where these oxytocin and vasopressin receptors have been found on the ligands. And then our approach is to search the literature, to mine genomes, freshly published genomes for these receptors, to look into venoms, and also to look into plants. No one ever looked into plants for these oxytocin vasopressin ligands. So this is work with my friend and colleague Christian Gruber from the Medicinal Chemistry University in Vienna. You can see here that the cysteines and the proline 7 and the glycines is highly conserved. And then you have this loop region, which is fairly flexible. So that really helps us to find different ligands that still bind to the human receptors and might have different selectivity profiles. So we pretty much accumulated all the original sequences and started synthesizing them and testing them over all 4 receptors. Another approach was the genome mining. So we decided to develop an algorithm to look for the pre-peptides of this oxytocin, and then whatever genomes become publicly available, we would screen for the oxytocin vasopressin ligands. We've applied this on 3 ant species, and we found oxytocin-like peptides in ants. Now ants, this is like another interesting story, I cannot go into details, but ants is a superorganism and have been described genetically into details. But oxytocin is a very social bonding hormone, and no one really looked into what oxytocin as a signal hormone does in a superstructural organism such as ant colonies. So coming back to the discovery approach, we synthesized only 10 of these oxytocin analogs, and already we found one selective human B1B agonist, and that's again so far the most potent B1B agonist to date. And then SUS3, back to the venom peptides of the cone cells. Here we screened a couple of venoms and we found some of these conopressin, and we particularly got interested in conopressin T because that was an antagonist. And this one showed antagonistic activity on the human B1A receptor. And through very short structure-activity relationships, we were able to identify an agonist to antagonist switch between the oxytocin and vasopressin to conopressin T. And we now use this C-terminal valine to induce these antagonists in our next generation design of V1A antagonists. And finally, we looked into plants, and this brought us back to Africa, where an old traditional herbal tea was given to pregnant women to induce labor. And most of you know that oxytocin still today is the ligand of choice to induce labor in women, in the 9th month. So we thought maybe there is oxytocin-like peptides in this tea, but we are even more interested because in order to make tea and to get the action, you boil water up to 90 degrees, and women drink it, and it induces labor. And the holy grail of peptide drug discovery is to find an orally available peptide drug. So that would be a really hot topic. And we found in this herbal tea extract these species called cyclotides. David Craig in Australia is like the leader in this field, and has done only NMR structure, etc. And we were not only able to find these cyclotides, but we were also able to find cyclotides that in one of these loops have oxytocin-like sequences in them. And we classically took the herbal extract, fractionated it, and then put these fractions on myometrium, pretty much like uterus contracting muscle cells, and we were able to identify a couple of compounds, cyclotides, that have uterus contracting ability. And then we found out the sequences, and then we pretty much designed from these oxytocin receptor loop sequences. We made a series of different normal peptides, and we were able to find out one which is selectively active for the human oxytocin receptor. So these studies are all really promising. We still don't know, plants obviously did not produce cyclotides to interact with human uterus. But what this study tells you is that plants work like combinatorial chemists. They have this really rigid scaffold, and then they mutate these loops. And we are now following this up, and we believe that they want to interact with insects as a defense mechanism, since the oxytocin vasopressin receptor system is so old and ancient and found in so many insect species, we believe that these plant peptides modulate insects by using these cyclopeptide scaffolds in their expression. Just quickly, an overview of how we do it. We synthesize them on peptide synthesizers, and then we have these 4 receptors on this flipper platform where we look into calcium signaling, and then once we have selective hits or hits in general, we follow them up with radioligand binding assays, cyclic AMP for the B2 receptors, this one does the same for calcium, and then the classic IP1. And then this is how we find out the mechanism as well as like the full selectivity. Coming back to selenium, we want to use selenium or diselenium to make more stable oxytocin analogs. And you can see here in serum plasma, the more redox stable you make these disulfide mimetics, the better their half-life. And as you can see here, the fire ether bond, which is non-reducible, has the best half-life. So the next step was obviously to move on and make the selenium ether bond, because the chemistry is just easier. The selenium is way more reactive, it displaces the chloride, and you can easily form these selenium ether bonds, and they completely isosteric to oxytocin, so they have the same activity and the same selectivity, but they are much stable in plasma. And we're also interested, since oxytocin is used to induce labor, particularly in Africa, a huge problem where you have problems with refrigeration. So these selenium ether analogs are much more stable at 55 degrees and will have a much higher shelf life than oxytocin itself. Another target we found was the oxytocin receptor in the gut was upregulated, and we were able to show that when we put our selenium ether analogs on these upregulated oxytocin receptor in the gut, it has analgesic effects. We were not sure yet what the mechanism is, but oxytocin receptor after this study became now a novel target for IBS and IBD, and selenium oxytocin analogs per se might not be able to be orally available, but we can graft or we can make these peptides into scaffolds that may be able to penetrate gastrointestinal receptors. Lots of people ask me about selenium toxicity, so I just put one slide up there. In general, selenium is good if it's in proper amounts. So there's selenium supplements, particularly as a healthy antioxidant. There's lots of studies showing that selenium is good against cancer, particularly since we have selenocysteines in selenium protein. So these are often redox molecules and pretty much help you against free radicals. And sources of selenium are usually grass nuts, fish, garlic, and meat. And very little is known of the acute toxicity, but guidelines say we give about 400 micrograms as a daily intake. That's okay. There's been 2 case studies. One was based on a mislabeling of the supplement, so they gave in like 400-fold more doses of selenium in these supplements, and then people who have been taking it for weeks saw that they were losing their hair and losing their fingernails. And that was because of a too high intake of selenium. And one person by accident took up 10 grams of selenite, and then he died after 6 months, 6 hours. That's a pretty sad story, but it's very hard to go 10 grams. So I did the calculation when it goes to therapeutic window. When we, for example, use our selenium oxytocin, we're about 10,000-fold away from that toxic drug dose. And if you would, for example, put in a disulfide bond to stabilize bietta, very hypothetical because it doesn't even have a disulfide bond, but just to see the dosage of these peptides are about 10 micrograms a day, and if we would put in a disulfide bond in there, it would still be 1,000-fold off that 400 micrograms a day, and that's very conservative, very low. So often the pharmaceutical industry say, like, no, we don't want to have any selenium in our drugs. I think that's it needs to be updated. We believe that selenium itself has good properties in peptide drug design as well as in small molecule drugs. I think excellent has been done. It's the closest small molecule drug that's nearly on the market in Japan. phase 3 clinical trials, and they don't really see toxic effects. To finish off, I just want to tell you what we do being an academic lab. So all these venom peptides and oxytocin ligands, we can modify, putting on fluorophores, near-infrared dyes to do 2-photon imaging. We can put biotins wherever we want them, or even like chelating agents such as DOTA for PET or MRI studies. So people, and this is why I'm here, in order to collaborate, or if they want to have novel tools, which are really highly potent and substance selectively, we can really modulate them to whatever applications you would need. And this could be like pharmacokinetics, using PET imaging, or tumor targeting, or just standard internalization, cell assays, or via fluorophores. We even use quantum dots. Here we don't label the peptides with quantum dots, but we label the quantum dots with peptides and try to direct quantum dots to whatever cell surface the receptor expresses. And with this, I'd like to conclude. I hope I could give you a little bit insights on the whole venom peptide drug discovery field and how we use selenocysteine to improve the discovery approach and particularly the structure-activity relationship studies. I hope I was able to show you that selenocysteine not only is a good folding catalyst, but can also be used for NMR, X-ray, or mass spectrometry studies. The disulfide bond itself is a little bit more hydrophobic than the disulfide bond, and we believe selenoether bonds from all the disulfide mimetics is so far the best in terms of isosteric as well as redox stable and thermal stable ones. And with that, I would like to thank all the people involved. I've been in 3 labs over the last 6 years. Most of this work originated where I did my PhD and my first postdoc with Professor Paul Aylward and Richard Lewis lab at the Institut for Molecular Bioscience in Brisbane. Then I moved to the Scripps Research Institute where I worked with Professor Phil Dawson, and now I'm with Professor Fernando Alvarez up in Barcelona. And these are my collaborators, Christian Gruber at the Medical University of Vienna, and BGG then today at the Institute for Neuroscience in Vienna. And with that, I would like to thank you for your attention. I'm more than happy to answer any questions.
Preguntas, questions, curiosities about the very impressive, utility short. That's very impressive. What about these venoms on the species that they produce? I mean, the conus, the snake. One snake bites another one. Are they sensitive to these venoms? Are they resistant?
So there's 3 different species. One are the fish hunters, which are the most interesting for us because fish receptors have like the most similarity to human receptors, particularly when it comes to ion channels. And then the ones that hunt mollusks, and that includes other species of constants. So there's whole common toxins, constant wars going on, and you see lots of video if you just go on YouTube where one constant hunts another constant, and then these constants defend each other. And that Nature Communication paper that came out was actually showing that these constants have access to different venom peptides repertoires if they want to defend themselves versus if they want to hunt. And then the last ones are just worm hunters. So yeah, a huge variety.
I'm surprised about that.
There's one species, the Conus geographus, which is actually lethal to people. So there's been fatalities, and you've seen these pictures where the cone shell is really nice. They have like these different patterns, and people collect these. And when you go out on the reef, the Great Barrier Reef, and you are from Europe and you're not familiar with the toxicity of the venom of these things, people go and collect these shells. And the first response this cones will have is like to retreat in the shell. So often it looks like it's already an empty shell. And then they put it in their pockets or keep them in their hand looking for more. And after a couple of minutes or hours, the console gets a little bit pissed off and comes out and stings. And if you happen to pick the wrong species, you can actually die. But most of these, when you go out of these research facilities, they will actually introduce you that you not pick up these ones. And this was these fatalities, I think, in the 60s or 70s, where the initial reason why Bandolero or Oliveira started studying these constructs, saying like, you know, if a snail can kill a human, something must be happening, and something active must be in there that acts on whatever receptors. And this pretty much opened the field of ion channels 30 years ago.
More curiosity, these consonants seem to be quite specialized, they have quite specialized what they are nothing. Why do they have thousands of different peptides to react?
So that prey is very specific, let's say fish. But the predators are not. And I think that diversity comes twofold. One is the way they produce these flavors. So you have to think about it like a combinatorial library. So these cysteine frameworks are highly conserved. So the scaffolds, there might be 20 to 50 different scaffolds, which are well conserved, not only within a species, but also interspecies. And the only thing that changes are the loops in the amino acid. So they have a cell machinery where they can produce thousands of compounds very easy and very rapidly. Yeah? And I think when it comes to evolution, how it's selected it's not because of what they prey, but the predators. Yeah? And there's different. They have to, since they live in the reef, they not only have to fight against fish or you know, whatever marine creatures hunt them, but also against land animals or humans who pick them up, or when there's low tide they are exposed to birds, etc. So I think the diversity of predators kind of like fuels that diversity of needing to have so many different subtypes and different components. And I think it's an obvious one after I talk, but a key misconception often when we think about using venoms or toxins to develop drugs. Because the first thing is toxic. But the toxicity or like the lethal problems are only derived because when a snake bites you or a spider injects its venom, there's like 1,000 compounds moving into the bloodstream. And that pain and the side effects come from the combination of all these compounds acting on your body. And when we design peptide therapeutics, we literally isolate one subtype selective compound, and then we use this one for therapeutic development, such as we can see with Grial, that is like a tight calcium channel blocker, and therefore you cut the pain signal.
Any other curiosity?
We are now in Barcelona, over Spain and Mediterranean.
No. So this is the big disappointing thing about the Venomics approach. I now, like 5 years ago, we would go to Papua New Guinea or the Great Barrier Reef or Borneo in the jungle, and it would be fantastic. Students loved it. It was always an adventure. Now I call my friend in Montpellier and say, "Do you have any new sequences?" And then he sends me an email with like 100 sequences, and that's it. And I'm busy for the next 2 years. So we still try to organize these field trips because I like the idea of still going out in the nature, because with that herbal extract will be found in Africa. I think traditional medicine or nature itself, by just observing them, still gives us the best ideas and the best sources of therapeutic drug development. But yeah, if you're a pharmaceutical company, for example, you would probably just use bioinformatics and transcriptomic approach to get sequences and make libraries.
Any other question? Curiosity? No? Okay, but now you know how venoms act and why.